These are your chapter 13 notes on sexual reproduction and meiosis. So kind of recall, maybe, hopefully, from Biology 1, that heredity is the passing of traits from parent to offspring. This is where we get words like inherit, or it's a heritable trait. Sexual reproduction creates variation, and when we mean variation, we mean genetic. There's lots of variety created from sexual reproduction. Why? Because when two different gametes have to come together, they mix that genetic information, and so no offspring will look exactly like either parent. Genes are inherited. Genes are segments of DNA that code for your traits. They actually code for amino acids, which form proteins. But ultimately, the proteins are what are going to make your traits or make you look the way you do. Gametes are sex cells. It's another term for them. You may also hear them called germ cells. These are egg and sperm in humans and many other animals as well. And actually, I think in all animals, I'm trying to think of an example where sperm and egg are not in the animal. I think that is the way. Chromosomes can be different, obviously. Not all species have X and Y, but and not all species does the male determine the sex, though in humans the male does. Gametes can also be other things. For example, in a flower, it could be an ovule and pollen. Doesn't necessarily have to be sperm and egg, but that would be in animals. Somatic cells are body cells. This is anything that is not responsible for reproduction. So a liver cell, a neuron, a skin cell, we could go on and on. Blood cell, muscle cell, on and on. So anything that's not responsible for reproduction is called a somatic cell. Asexual reproduction gives rise to a clone. So that means that it's going to be exactly identical in appearance, with the exception of mutations. So if there's a mutation in there, then that may mean that they're not going to be identical. Mitosis is technically, we could say it's a form of asexual reproduction because your cells are reproducing and they're producing cells that are identical to the originals. Sexual reproduction involves two parents giving rise to an offspring that is genetically a combination of the two, and again, that creates variation. Why is that important? Well, having a lot of variation means that you're going to be more likely to survive if the population if the environment changes. So kind of an example of this would be the bubonic plague. It killed a lot of people, but it didn't kill everybody because not everybody's immune system was the same. If everybody's immune system had been the same and that immune system was susceptible to that plague and it killed that person, it would kill everybody. So that also facilitates adaptation. So traits that are favorable to the environment. Traits that are going to be good when times get hard. It speeds up evolution. Again, that's going back to the variation and the idea that the more variety there is when the environment changes, more than likely somebody in that population is going to survive, and they're going to be able to reproduce and pass on their traits to their offspring. And those traits are obviously favorable to whatever that environment is because they were able to survive it. The disadvantages of sexual reproduction is that it costs a lot of energy because it usually involves more than just a cell dividing. There's two gametes that have to come together, which means there's energy in that, plus the energy in creating the gametes, creating the egg or creating the sperm. A lot of times when you're talking about like animals, you're talking about a courtship, time, resource consuming for that to happen, just putting that into effort. And it usually sacrifices the fitness of one sex to the other. And so there's there's a disparity there between the sexes. Um, Asexual reproduction kind of is almost like a flip-flop. Not a lot of energy expended in this because you're just dividing a cell. No courtship because there's only one parent involved or one cell involved. And there's a great increase in fitness for each individual. There's not really a disparity between individuals because they all reproduce the same way. Excuse me. The disadvantages is that there's not a lot of genetic variation, which means that the environment changes. They're all going. Adaptation to the environment is difficult. Usually can occur through mutations. But aside from that, there's not much that's going to make them more varied. And retards evolution, which basically means it slows it down. It makes it so that it doesn't happen as quickly. Although, bacteria have evolved pretty rapidly, considering that it's asexual reproduction, and probably, even though we say that it slows it down, the reason bacteria probably evolved so quickly is that they reproduce very quickly. You can get a new bacteria about every 20 minutes, depending on the bacterial species. All right, chromosomes. Autosomes are the chromosomes that are not your sex chromosomes. So we have 23 pairs of chromosomes. 22 of them are autosomes. The 23rd pair, which is right down here, are your sex chromosomes. That's going to determine whether you are male or female. XX is female and XY is male. And you can't see what it says at the bottom here, but it probably says, note, not all species are the same. For example, females are XX in humans and XY are males in humans. But I think like chickens, there's like a Z chromosome. It's not even a Y. I'm trying to remember all the different species I've learned over the years. But not even all of them use X's and Y's. And some of them, the male has the two that are the same, and then the female has the two that are different. It just varies species to species. A karyotype is a photograph of chromosomes. It indicate the correct number of chromosomes, gender, correct length, not specifics, but large-scale differences. So karyotyping can be done, for example, to look for Down syndrome, because Down syndrome is a trisomy 21, which means the 21st pair of chromosomes, there are 3 of them instead of 2. So I'll go back to this diagram real quick. So 21st pair right here. So Down syndrome would be having 3 of those instead of 2. It's one of the only trisomies that actually results in a live birth and a functional individual, although they do have mental retardation. Trisomy 18 right here, that actually develops into Edwards syndrome, which is catastrophic for the baby. The infant can be born but will not usually survive past a couple of months. So here's a picture of kind of what they do. They stain and photograph the chromosomes, and then they kind of cut them out. I don't know if they still cut them out. They probably do something fancier with technology and arranging them. But they arrange them according to their size, and the largest is number 1, and then from there on. And a normal individual should have 22 pairs plus the 23rd pair, so a total of 46 chromosomes. If they had trisomy 21, they would actually have 47 chromosomes. Female karyotype, and then male karyotype. And this is a haploid karyotype. The difference here is that they're not showing both pairs. So here they've got 1 pair and 1 pair. One came from mom, one came from dad. Here they're not. So this would be like a sex cell that is haploid. So this would be like a sperm for a male and an egg for a female. So we did this in class, but haploid versus diploid. Diploid cells contain 2 sets of chromosome, 1 from each parent. All your somatic cells are going to be diploid, which is 2N. We did practice this in class, and you guys did great, so I'm going to kind of speed through this. Haploid cells contain 1 set. Gametes are haploid. These are going to be the only cells in your body you're going to find that are haploid. That would be in all animals. In, yeast, they can take either a haploid stage or a diploid, maybe depending on your environment. So there are organisms that can kind of fluctuate between both of those. I think your textbook shows life cycles of several different common organisms, like maybe, I think, Rachel, mold, and yeast, and because some of them are a little different in how they alternate between haploid and diploid. You don't have to know all those. Okay, so figure 13.6 in your textbook is what I was just talking about. It shows you a variety of sexual life cycles where they alternate between haploid and diploid. One example is alternation of generations. It alternates between haploid and diploid. They're not really making egg and sperm that are combined through sex, but their generations alternate between a haploid state and a diploid state, and their cells can combine when they're in a haploid state to form diploid, which can then divide through mitosis. All sexual life cycles share one thing in common. They have genetic variation because you're combining two haploid cells that are genetically different, and you're getting a diploid cell that has a combination of both of those genetics. So here's a plant life cycle, an alternation of generations. So it shows you where the haploid is, and then where the diploid is here. So fertilization, zygote, the embryo is developed by mitosis, just normal cell division. Creates a sporocyte. That sporocyte can then undergo meiosis and produce spores, and then spores can actually combine, and they actually call them egg and sperm here, though I don't know that they're really technically what we would see as egg or sperm. They don't have little tails or anything. And then this is just another one, plasmogamy. Not even really sure what it is, just kind of showing it to you to show that there are other options. Apparently these can go through sexual reproduction, but they can also go through asexual germination, which tells me it's some kind of a seed. It's got a spore. A little bit about the human life cycle, because that's the one we talk about the most. Fertilization creates the zygote. Zygotes continue to divide through mitosis to make more cells. Only cells in the human body that are not produced through mitosis are the gametes. Gametes are produced from the gonads, ovaries or testes, and they produce haploid cells. Everything else is diploid. Even the zygote is diploid, because the zygote is formed from a haploid egg and a haploid sperm. Here is the human life cycle. Sperm meets egg, 2 haploid gametes, fertilization creates a diploid zygote, continuous development through mitosis until the organism grows up. All the cells of the human body are diploid. The gonads here, the ovary or the testes, are going to produce sperm or egg, and then that process is known as meiosis, and then those sperm and egg will again combine into the next generation. Meiosis is the gamete, or actually it's pronounced meiosis, I apologize. The gamete formation comes about through meiosis, slightly different process in males versus females. That's called gametogenesis, how it's different. Meiosis is a cell division that reduces the number of chromosomes from diploid to haploid in the gametes. Fertilization then restores the diploid number when egg and sperm combine, 1n plus 1n equals 2n. So how are they different? This would be male, and this would be female. So in males, the cell goes through meiosis, and then it produces 4 cells, and those 4 cells gain a tail, and the head is changed in shape, and so on and so forth. But ultimately, the 4 cells produce 4 sperm. In females, the egg undergoes meiosis, and then what happens is one of the eggs is kind of recruited to become bigger and better, and the other one is what we call a polar body, and it just disintegrates. And then this egg is just nourished and flourished and becomes very large. And so in comparison, if you look at the sizes, the egg is actually much, much larger. Females are also born with all the eggs they will ever own. Males produce, I don't know what the statistics is, but lots of sperm daily past sexual maturity. So that's a little bit of how that's different too. I like to say that way not politically correct, but I like to say that males, sperm are not really high quality. A lot of sperm have 2 tails, 2 heads. They can't swim in the correct direction. They swim in circles. So men, because their sperm are not quality, they're shooting for quantity. So they get 4. Females only get 1 egg for every round of meiosis. But it's a really high quality egg. It's usually a very good, and as long as it is ovulated before 40 or so, it's probably going to be really good. And even after 40, the egg can still be good. It's just that the quality diminishes a little bit after, I think, about 35, which is why you get more incidence of Down syndrome. Meiosis, 2 stages of cell division. Meiosis 1 reduces it's called reductional division because it reduces the chromosome number. Because you're going, in meiosis, you're going from 2N to N. And then the second stage actually just evenly divides what is there into the 4 cells. Distinct features of meiosis, crossing over. We're going to talk more about this a little bit later. We're going to model it in class, but we're going to do some like calculating how often crossover happens and stuff in genetics. The chiasma, I'm hoping I'm pronouncing that right, is the crossing over region. It's where the crossing over is happening. In any 2 given homologous pair of chromosomes, crossing over can happen here, it can happen here, it can happen here. It can happen, many spots on any 2 homologous chromosomes. Homologous chromosomes are going to pair up in metaphase 1, and then they're going to separate in anaphase 1. Let's take a look at what this looks like. So here is a pair. Now you would have 23 pairs. Okay, keep that in mind. This is just 1 pair, just to give you the idea of what's happening to each of the chromosomes. So in interphase, remember in the S phase, the DNA duplicates. So that's why it's showing here a duplicated chromosome and here a duplicated chromosomes. So these are going to pair up, and then they're going to separate. The homologous pairs will separate first, and then the actual chromosomes will separate next, so that each cell is getting one of those. Now, obviously, in humans, there would be 23 in each of these. Non-disjunction is something that can happen in meiosis that can actually create a problem. It's mentioned a little in a later chapter too. Chromosomes don't separate properly in meiosis. So instead of separating out like this, maybe this one went over here and this one has none. Or it's missing one. If you're doing 23 pairs, it's unlikely that all 23 would be wrong. Or maybe over here at this stage, both of these chromosomes went over there. And so now you have extra ones in both of those. It results in aneuploidy, which means an incorrect number of chromosomes in the egg or the sperm. Monosomic means you're missing 1 chromosome. Trisomic means an extra chromosome. Remember we talked about Down syndrome being a trisomy. Polyploidy, an entire set of chromosomes, usually only happens in plants. That would be not happen in a human and actually result in a live child. So here's an example I kind of talked about. Here your pairs are supposed to separate out. Well, they don't. So now all of a sudden these sperm have extra and these have none. And this is showing here where they separate out normally here, but then when this is supposed to split, it doesn't, or this one's supposed to split, and all of a sudden you have 2 here, and then these only have 1. And so it can get kind of interesting the way those can divide out. And there's lots of disorders that are associated with that. And this is Down syndrome. Just kind of on a side note, interestingly, if this happens to the sex chromosomes, you can get things like females that are XXX. You can get males that are XXY. This is called Klinefelter syndrome. You can get X0. So basically you can get 1 X for the female and then nothing else. No Y, no X, nothing else. This is called Turner syndrome. So there's some really interesting things you can get with, and most of those actually do develop. Normal individuals are sterile, so they can't have children of their own, but they do develop usually, sometimes, well, most of them I think have normal intelligence. There are a few of them that I think may have a little bit of maybe some learning disabilities or something, but all develop perfectly, grow into adults. Sometimes don't even catch this until, late teenage years, 20s even. Sexual reproduction contributes to genetic variation. I mentioned that earlier. How does it do that? There's 3 important things. Independent assortment. So when all those homologous pairs line up randomly in metaphase of meiosis 1. They do so randomly, and so it depends on which side. Here's a one, and this is a different one. Which side of the cell they line up on depends on which ones are going to go together. So in this case, these three went together, and these three went together. But I could have just as easily said, well, this one lined up here, and this one lined up here, and this dark one lined up here, and this one lined up, and this dark one lined up here, and this one, and then these three chromosomes went together, and these three. So this is how, like, even though your parents have lots of children, if your parents have 3 or 4 children or lots of children, none of them look exactly alike, because when this happens, you never know what you're going to get. One child might look more like mom, and one child might look more like dad, just because of the chromosomes that they got. Crossing over creates recombinant chromosomes by exchanging DNA. We're going to look at that here in a little bit. And then the fact that it's random fertilization. No 2 eggs and no 2 sperm are alike. And so every time they combine, it's going to create something different. This is crossing over here. So these chromosomes are really close to each other in prophase 1. This happens in prophase 1. And so when they do that, they actually exchange chunks of DNA. So this is also why you can look more like one parent than the other when technically you have chromosomes from each of your parents. You have 23 from each. Is because this might be, mom's chromosome, but it's got a little bit of dad's DNA on it now because they're sharing that genetic information. So this shows independent assortment, what I was just talking about before. These are the different options you could get depending on where those chromosomes line up. Now, this is only showing 2 pairs. We have 23 pairs of chromosomes. So when you arrange 23 different ways, you can get so many different options. I tell my students, think about it like if I ask them to pick a partner and then line up in a line at the door, and I have 23 pairs. Well, that means I'd have to have 46 students in the classroom to begin with. That would be pretty hard. But I tell them if they paired up and they all lined up, and then I told them to do it again but stay with the same partner, they would never necessarily be on the same side. You would always get different results when they separated from their partner into different cells or different groups. I don't know if that's easy to follow or not. In my mind, I see it and I think that makes sense, but I don't know if it really does. So the evolutionary significance of those that are best suited to the environment will survive better. So if you have traits that are favorable in that environment, you'll survive better. Natural selection results in the accumulation of genetic variations favored by the environment. So why armadillos have all kinds of armor? Because having armor helped them survive. And then when they survive, they reproduce and they pass on that armor's trait, the armor, that body shielding armor, to their offspring. And so over time, natural selection has favored that particular genetic variation. And that works for anything. That was just my example. All right, that's the end of that. Go back and review meiosis. If you need to watch Bozeman's video, do that. I'm not going to go through specifically all the stages of meiosis, but it is meiosis 1 and meiosis 2, and it goes through prophase 1, metaphase 1, anaphase 1, telophase 1, cytokinesis, and then prophase 2, metaphase 2, anaphase 2, telophase 2, cytokinesis. Very similar things happening. We're going to do a little drawing in class, a little modeling. It's kind of like mitosis, but it's happening again a second time because they're pairing up in pairs again in metaphase, and they didn't pair up before. In mitosis, they all just line up in one single fat line. In meiosis, in metaphase, they line up in pairs, so the pairs split up first, and then the chromosomes split up, and that's how you get them into haploid versus diploid. So take a look at his videos or go online and just Google search meiosis animations, and there's tons of them out there. I'm sure I'll show one in class. All right, have a good night.